My name is Daniel Burkhart. I'm a sixth-year PhD student in genetics, and I'm going to talk to you about some methods that, bulk of my talk is going to talk about this problem of quantifying the effect of experimental perturbations in single-cell data sets. And so, the reason we're all here talking about single-cell data is sort of an advance that happened in around 2015. This allowed us to measure RNA and many other kinds of features in individual cells at unprecedented scale and resolution. And I always sort of like to start talks looking at this image. If you haven't seen this, this is a bead and an individual cell coming up from the bottom, getting captured in a microfluidic droplet that we can then use to generate single cell libraries of RNA. And the result of these kinds of experiments are really large data sets where we have either some sort of set of genes or proteins, anywhere from dozens to tens of thousands of features. We have anywhere from thousands to sometimes millions of observations. And then we also are starting to collect data from multiple kinds of samples. And a lot of the methods that we work on in the Krishnaswamy lab and the methods I'm going to talk to you about today are all sort of motivated by this general idea of trying to identify and learn structure in high dimensional data. We have some set of measurements here. I'm showing 3 dimensions. We were talking about maybe having tens of thousands of dimensions, but we make some assumptions that the data doesn't actually sort of uniformly inhabit this ambient space, but actually lies in some low dimensional latent structure that is smoothly varying and locally Euclidean, but maybe not globally Euclidean. So you might not be able to make jumps where there's no data, but you can walk through the data to get to similar observations. And the kinds of computational methods that we use to learn that underlying latent structure in the data is called manifold learning. And the general idea behind manifold learning is you have data in some set, kind of ambient dimensions. Here I'm just showing 2. You can look at distances between all points of cells, and we see that there are some spurious connections here. We only want connections along this path, but we can use certain kinds of functions, like a kernel function that takes our distances, build some sort of affinity graph from them, where only local relationships are preserved. And then we can perform certain kinds of operations over those local relationships, like some sort of data diffusion, to actually identify underlying structure and geometry of the data set. And when I say geometry, I mean we want to know how far is this cell from this cell, and it's not actually just the distance in X and Y here. We actually know, because of this manifold learning, that you have to walk around the data set to actually identify that true underlying distance in the data set. We can then use this geometry to form a bunch of different kinds of downstream analytics tasks. We can do clustering, we can do dimensionality reduction for visualization, we can do kinds of data denoising tasks based off of this. It's really a fundamental idea that powers a large diversity of algorithms. I'm going to sort of touch briefly upon 2 of them, FATE and AANet, and then spend most of the talk talking about MELD. Many of you are probably familiar with FATE. We're talking about dimensionality reduction for this data, so you can actually look at it. A sort of classical way to look at high dimensional data is just to do principal components analysis, where you look at a linear combination of the features that identifies maximum variance. This is a data set of stem cells grown as 3-dimensional embryoid bodies over a time course. The color, each point is a cell. The color represents the day that the cells were sampled from. We sort of combine some time points. What you can see is that PCA identifies some global structure in this data, right? We have early cells have low loadings on PC1. Cells that are later in development are a little bit farther to the right, and there's some smooth variation between them. But there's 30,000 cells here. It'd be really hard to identify the biological heterogeneity. Thankfully, there's a large number of algorithms that have been developed to do this kind of task of taking 10,000, 30,000 dimensions and reducing them into 2. Here I'm showing 6 of them. Many of you are probably familiar, if you're interested in single cell, with TSNE or UMAP. If you're familiar with our lab, that we can look at methods like FATE. And the main thing I wanted to communicate here is that sort of these 2 methods, I think, would sort of qualify as both manifold-based methods. And what you really get when you use this paradigm to design methods is that we go from sort of these blob, sort of coarse-grained, difficult to understand structures especially with fate here, you can see we can actually preserve global structure within the data, but also resolve some fine-tuned local heterogeneity and actually capture the sort of long-range connections without shattering them, where some methods might, like UMAP might, preserve some of that local structure, t-SNE preserves some of that local structure, but loses those global connections. And this really aids in the analysis and exploration of high-dimensional data. Another way we might use this manifold concept to analyze single-cell data is using a paradigm called archetypal analysis. So many of you are probably familiar with the idea of clustering, where you take a data set, you break it up into a number of discrete partitions, and you say a cell is either in cluster A, and then you say cluster A is T cells, and other cells in cluster B, and cluster B is natural killer cells. But we actually understand that there's some more fluid continuum between them, right? We often think of biology as a spectrum, and archetypal analysis is another approach to characterizing biological heterogeneity that also takes this kind of spectrum idea in the data, and instead of just saying you are one cell type, you're one cluster or another, represents each point. So here we're looking at microarray data, and you describe it as close to or a combination of several extrema or archetypal data states. These archetypal states, in immunology might be exhausted T cell versus naive T cell versus effector versus helper or regulatory, and you're somewhere in between that spectrum. Classic way to do this is just in a linear way in the ambient feature space. But one of the techniques that we've developed in the Krishnaswamy lab is a method called archetypal analysis network, which is a neural network, which takes some sort of nonlinear data in the ambient space, like we observe in biology, and uses a neural network autoencoder model with some novel regularizations on the latent space to enforce it to have some of the things we would like to see based off of this idea of an underlying manifold of the data. We want to be able to interpolate between points reasonably. We want to make sure that each point is representable as a convex combination of each of these archetypes. But we also want to be able to say there might be some nonlinearity in the relationship between the feature space and the latent space. And I understand I'm going very quickly through these methods to sort of explain exactly how they work would, I think, just take a lot of time. But I want to just maybe get across this idea that this one underlying idea of thinking about single cell data as originating from a manifold is very powerful for a bunch of different kinds of analytic tasks you might want to do. So that I'm going to spend the rest of this talk talking about single cell RNA sequencing data used in the context of experimental perturbations. So, sort of the first set of single cell experiments that were done, sort of right after 2015 with these droplet techniques, was just characterizing biological heterogeneity. But as time has gone on, people are starting to actually use single cell experiments as an experimental readout. So what I mean by that, we're looking at a paradigm here from a Crispr screen experiment published in Nature Methods in 2017. You might take some kind of T cells, split them into two different groups, a control group and a treatment group. The control group, you maybe just add some media, collect, and then collect single cells and then sequence them. In the treatment group, what they did was use anti-CD3, CD28 beads, which simulate the T cell receptor activation pathway, and then now have this treatment group. And the result is you get out a single cell matrix where you have a bunch of cells and the genes that they're expressing, but you also know the condition that they were from because we collected two different set of libraries. And you might want to know, okay, what's the difference between these two data sets? What was the effect of adding these anti-CD3, CD28 beads? So based off of the way we often think about bulk RNA sequencing, you might want to think about just doing a standard differential expression test. We have two collections of samples. There's maybe 3,000 observations in one sample, 3,000 observations in another. This is a really large N. We can now start to actually do sort of mean testing, sort of your standard differential expression test, Wilcoxon t-test. You can use a wald test like in DE-seq. But what we see when we just group the cells based off of the sample that they were from, and we look at things like expression of their genes, here we have each point being a cell. The position of the cell on the x-axis is the condition that they came from. The y-axis is expression. And we're looking here at 6 different genes known to play a role in the T cell receptor activation pathway. The red dots here represent the mean expression in the samples. we can see that even though we know these genes either should go up or down, if we just look at the means, the difference is very, very small. And standard differential expression tests are not going to give you a really good set of signatures or understanding of what the effect of that perturbation was. Another way you might think about doing this, and this figure is from the Dattlinger et al. Paper, is look at some dimensionality reduced representation of the data. I was talking about PCA earlier. You might want to look at the first two principal components of the data and see where's the separation between the samples. And the answer is that there's actually not a lot of separation of the samples. So again, each point is a cell here. What we see is right there, right above my cursor, there is a green cell from the naive condition that's surrounded by a bunch of cells from the treatment condition. Similarly, if we look on the left, we see that there are a large number of blue cells here from the stimulated condition that, at least along the axes of maximum variance in the data, are very transcriptionally similar to cells that receive no perturbation at all. And this is actually why the difference in means is very similar. Transcriptionally, at a global scale, there's not a lot of difference between cells that were stimulated and not stimulated. There may only be a handful of genes that are affected, but the global biological heterogeneity in the data set and technical noise, just from the fact that we are only sampling, say, 10% of the RNA molecules in a cell, means that the effect size of our perturbations are actually relatively small with respect to the biological and technical noise. And so simply doing a global comparison, taking all cells from both data sets and comparing them, is not going to give you a reliable estimate of the effect of the perturbation. And so a couple of years ago, started to look at this kind of data and really started to take a step back. What do we want to do when we're taking single cell data sets and using these as a measure of the effect of a perturbation? And the way that we actually like to think about this is to have some underlying manifold model of the data where there's some underlying state space. So we're talking about not necessarily 10,000 free axes of variation, but maybe only several hundred. And there's some underlying density over the data set. And if you have some control condition, there's a density function over this underlying state space that dictates the frequency with which you'll observe any given kind of cell, collection of UMI counts in that data set. So we would expect to see maybe a lot of cells of this state, a lot of cells of this state, and not many of cells sort of in the back here. And one of the key things that we assume is that if you go and just change a gene a little bit, the probability of observing it is not going to change that much. We call that locally smooth or continuous in the data space. And this is sort of the underlying idea of the manifold model, is that if you change things in the feature space, like in the gene space, just a little bit, the underlying biology is not going to change that much. There may be slightly medium scale changes that have huge differences, but not at least, change one UMI count. So the effect of the perturbation here is actually, we think, to change this underlying data density. So you add a drug, knock out a gene, turn on a gene, change the growth media. And what's going to happen is that some kinds of cells are going to be more likely to be observed. Some cells are going to be less likely to be observed. And some cells are not going to change at all. And so what we really would like to do, if we're trying to quantify the effect of a perturbation, is to calculate the change in density over this underlying state space that tells us for any given possible combination of gene expression values, what's the change in density that we would observe? Or is this, particular kind of cell or collection of cells enriched? Is it depleted, unaffected, et cetera? And so those of you who are familiar with some ideas in statistics might be somewhat familiar with this idea of a kernel density estimate, or a KDE. If you think about data here being, this is just a random sample from two distributions. We're just looking at one feature here. And this is the number of samples we observe, right? Just imagine taking 500 samples from some random variable or variables. The idea behind kernel density estimation is to take a function of this input data and some bandwidth parameter t, and produce some smooth estimate over the underlying feature space. Here, we're just looking at the number line, basically, and to give you a density estimate of the probability of observing any one of those numbers, right? So I'm saying we see a lot of observations that are right around 2. So the kernel density estimate of the data at 2 is high, and especially it's higher than at seeing something at around 0, for example, because we observe more data observations here. And I'm not going to spend too much time going into the math. I think this audience has a lot of biologists. I'm happy to talk more if you have questions at the end. I'm also going to introduce this algorithm and then pause for people to ask questions. But the basic idea is we have some function that takes in real data and gives us a kernel density estimate. This is in real in the space of real numbers. There's a problem with this approach, though, for single cell data. Doing kernel density estimates in high dimensions is incredibly challenging because the number of observations that you need scales exponentially with the number of dimensions that you're trying to estimate. So if you want to estimate just in one dimension, like I showed you before, you actually don't need that many observations to get a reasonable estimate of the density of the data. But if you scale the dimensionality even just by a few genes or features, all of a sudden you get an explosion in the number of observations that you need. And so the solution to this problem is that instead of just taking the kernel density estimate in the feature space, what we do is take a lower dimensional approximation of the data using this manifold model to build a graph that we can then use to approximate the lower dimensional state space and just calculate the density at each point that we observe. So I say, for a given cell that we do observe, I could say, what's the probability that we would see that cell in one sample or in another sample? But now there's sort of a different problem here. The function that I showed on this slide requires a kernel function that's defined over the domain of the data. Here I'm looking at the space of real numbers. But once we start to do this estimation over a graph, we're no longer looking at real numbers. We have a discrete space. And so we need to have a new kind of kernel that we can then use to estimate the density of each of the samples. And so the way that we ended up doing this is actually use the eigenvectors of the graph Laplacian as a kernel support. And I know there's a lot of you who probably have no idea what a graph Laplacian is. I just want to assure you that this is a way to use some intrinsic features of the graph itself to approximate a support for our kernel density estimate. And to give you an example of how this algorithm works, I'm just going to show you some cartoon data or some cartoon examples. We have our control and treatment experiment. We get our two data matrices, our two conditions out here. And the first step that we do is we create a cell similarity graph. So you may have heard the phrase KNN graph, where you just take each node in this graph as a cell, and the edges in the graph are representing connections between cells. There may be some of you who hear graph and they think like a XY plot. There's a meaning in mathematics of a graph as a discrete model of data that has nodes, a collection of nodes and edges. I encourage you to do some reading on this. But the basic idea is we take our observations, we build we use a Gaussian kernel in the ambient space to build this graph. And the idea is that this graph approximates the underlying data manifold. And then what we do is we take our sample labels that say this is a control cell or this is a treatment cell, and we create a set of indicator vectors over the data set. And so we're saying now we have a graph, and what I'm doing is saying for each node in this graph, for each cell in this graph, I'm going to give that cell a value of 1 if it was seen in, for example, here are the treatment condition, and 0 if not. And so for a 2-sample case, we have 2 sample indicators. There's going to be a second signal that's all these zeros turn to 1 and all the 1s turn to 0. I'm not going to show you that for just clarity right now. Sorry, I guess this figure got a little bit mashed. Let me just take a note to clear that up. What we want to do is actually take a kernel over the graph and create some density estimate of this signal. So we're now using a graph signal, a vector over the graph. We're smoothing it over the graph, and we get some density estimate of that sample over the data set that we've observed. Now to show you how this works on real data, I'm looking again, this is the same set of T cells from that CRISPR screening experiment I was talking about at the beginning. We have cells that were stimulated with anti-CD3, CD28 beads. We have cells that are unstimulated. We know a set of cell barcodes, and then we have some sample indicator vectors that are 1 for cells from a stimulated condition, and zero otherwise. And then we have another indicator vector that's 0 for the unstimulated condition and 1 otherwise. And here we're just showing them on a fate embedding of the data set. Imagine this is very similar to the PCA plot I was showing you earlier, just using fate. And the first step is that we take these indicator vectors and we normalize them by the number of cells per sample, because there's no guarantee that you actually have the same number of cells in your treatment and control condition. And what this essentially gives you is the empirical probability of the sample on the data. So this is sort of, you were just to say, the probability of observing that cell, given that you are from this data set, is whatever is this normalized indicator signal. You can see that it's different for each of them, but these columns will now sum to 1. And then what we do is use a graph kernel that I was talking about before, a new kernel function defined over the graph. So here we're using f hat to define this kernel, where we're taking a set of data points on a graph S and some bandwidth. And then what we're going to do is actually create a smooth density estimate of the sample over the data set. Just sort of walk you through a little bit of the nomenclature down here. What we have here is some kernel function that's defined over the data. This function, what it does is take each cell and all of the other cells in the data set, and then uses the eigenbasis of the graph Laplacian to look essentially at local neighborhoods around that cell and say, what's the ratio of cells from one condition or the other. And so these are the sort of terms I was talking about using the Laplacian eigenbasis. These are the terms that are actually defining that. I'm bringing this up now mostly for people who are interested in looking at the manuscript, where we go into much more detail about how this kernel is derived. But this is a section of the presentation that corresponds to some more of those technical details in the manuscript. But the result is that we get some density estimate of the signal over the graph that's smooth. So cells that are close by, here I'm showing this on fate, but close by on the graph, have similar values of density. And then what we can do is we do this for each sample independently. So we smooth the data, we get a density estimate of the stimulated condition over the graph, we get a density estimate of the unstimulated condition over the graph. And then what we do is look at the ratio between them. So these two figures sort of correspond to those like control and perturbed landscapes that I was showing earlier. And what we can do is actually look at the ratio or the L1 norm of them, make each row sum to 1. And what we get is a likelihood. So these are probabilities. This is like sort of the estimated probability or the probability estimate, the kernel density estimate of the sample over the data set. But now what we're doing is sort of looking at the likelihoods. So we're saying, what's the likelihood that if you were to see this cell again, or let's say you saw a cell and you actually didn't have the label for it, you didn't know what sample it actually came from, what's the likelihood that that cell came from the stimulated or unstimulated condition? And this gives us the output of the MELD algorithm, which is the sample associated relative likelihood. And the rest of this talk, I'm going to talk about how you can use this sample associated relative likelihood to do all sorts of kinds of computational analysis to understand how these experiments work. But I really would like to take a second right now for people, feel free to unmute yourself, ask any questions about really anything that I've talked to up until this point. I know that that's like, for some of you, it's going to be a lot of new technical information. Happy to answer any questions. I'm going to wait at least a minute before I think I go on. So please ask questions.
Hey, Dan, Samit here.
Hi, Samit, how's it going?
Good. So I understand that you take a ratio here.
Yes.
But that's assuming there are only 2 conditions. What if there are more than 2 conditions?
Yeah, that's a great question. So I am going to show sort of towards the end an example of this if you have more than 2 conditions, but you take the same ratio just across all of the conditions. We're looking at them all to normalize to 1. And so if you had 3 conditions, the difference in probabilities is not going to be, so right now 0.5 means equally likely to be seen in any condition. If you had 3 conditions, 0.33 would be equally likely to observe in any condition. So we are always estimating the kernel density estimate per sample. and then we normalize across all of the conditions to get the estimate of the to get the final sample associated or condition associated relative likelihood. Does that make sense?
Yes, it does. Thank you.
Yeah, so you just add a column here and then you pass that into the L1 norm. Any other questions? I'm sure that there are some more. I'm certain that not everyone understands everything, so I'd love to help clarify what we're doing computationally before we go on.
Just one more question. So you said that you are calculating this kernel density over each sample. You are calculating it over each sample or every condition?
So the density estimate is calculated per sample. So all of the data sets are concatenated together. One of the assumptions is that you can learn a joint graph between all of the samples. And so here, I think you'll see it'll make it a little bit clearer. I'm going to show some examples where you have replicates of two conditions. And I think that might clarify like exactly what that looks like. But you do this per sample, and then at the end, you want one likelihood per condition, which might be the average of the replicates.
Okay, thanks. Got it.
Yep, no worries. It's difficult with no faces. I can't tell who's confused or who's not. I'm sure some people have questions. I'll give it a few seconds before I move on. Oh. Okay. Great. So at this point, I hope everyone is comfortable with the idea that we have some estimate of the likelihood that a cell is going to be observed in one condition or another. And here I'm showing you, again, this T-cell data set from, again, that same experiment I was talking about before, simulated, unstimulated. The result after you run MELD is to get a continuous spectrum of TCR induction. So what you see is that cells that are surrounded by cells from the treatment condition have high likelihoods of being seen in the stimulated condition. And cells that have really low, are mostly from the unstimulated condition or surrounded by cells from the unstimulated condition have really low likelihoods of being observed in the unstimulated or in the stimulated condition. If you're surrounded by cells that are unstimulated, you're unlikely to be observed in a stimulated condition. And then cells, you see sort of where there's a lot of mixing, we see cells sort of in the middle here. A lot of times, so one of the advantages of this method is that now for every single cell, we have an estimate of the effect of the perturbation. And I'm going to show why it's crucial to have this per cell. That being said, a lot of kinds of questions that people ask rely on some partitioning of the data. People want to have some set of clusters. They want to know what exactly is the population that is enriched or that is depleted. And I'll show a little bit later about why doing this just based off of the data alone, just taking your data set, running Louvain or k-means, and then getting your clusters, and then quantifying the effect of the perturbation is problematic. But once you've quantified the effect of the perturbation per cell, we can actually leverage this information to create a new partition of the data that gives you more meaningful information about the cells that are enriched and depleted. So here I'm just showing you sort of a cartoon example where we have some cells where there's a transition between some cells that are enriched in the control condition and enriched in the treatment condition, and some cells are just sort of completely mixed between the two of them. One of the things that we introduce in this manuscript is a method called vertex frequency clustering, which uses the labels, the sample labels over the data set, and the sample associated relative likelihood scores that we get from the MELD algorithm to create a new set of clustering that allows you to identify cells that are unaffected, in between being enriched or depleted, highly enriched or highly depleted. And there's a couple of interesting things I just want to emphasize here. So, we expect that the cells that are completely unaffected by the perturbation, or that are halfway in between a population that's going from enriched to depleted, are both going to have likelihoods of 0.5. You're equally likely to see them in either condition because they're either an intermediate state or they're completely unaffected. But you might want to know, for example, is a cell actually unaffected or is it is it halfway in this sort of intermediate state? And the way that we can do this is actually look at some spectral properties of the sample labels over the data set using a tool called the window graph Fourier transform. I'm going to maybe skip a little bit over how this works, but the basic idea is that we can use some information, that we can actually, there's information encoded in this data set that tells us that these cells are halfway in between these two conditions and that these are totally uniformly mixed. And that has to do with sort of the likelihood that a cell's neighbors or their neighbors' neighbors have the same label as that cell. And that allows us to create new clusterings of the data that we can then use to say these cells are enriched or not enriched. So as an example of how this is really helpful, one of the things that we did on this T-cell experiment was to use vertex frequency clustering to partition the data into activated, intermediate, or naive cells, and then perform gene set enrichment analysis using the same differential expression test, just between this activated and naive state, using either the sample labels themselves. This is just that direct two sample differential expression comparison I showed on like the second slide of this section, or a large number of other clustering algorithms that are just based off of the data geometry. And here we're looking at a number of Go terms that we know are implicated in T-cell receptor activation. These should be enriched in this experiment that we're looking at here. And what I hope is pretty clear is that the enrichment, the combined score, the level of enrichment for these gene sets is higher when you use these set of clusters than just comparing the samples alone or using almost any other kind of clustering algorithm that we compared here. This is, biology is difficult because you don't necessarily have ground truth. One of the things that we also wanted to do was take some computational simulated comparisons to sort of show that that both algorithms, MELD and vertex frequency clustering, work well. The basic idea here is to take either a set of simulated data sets using splatter where we know the underlying geometry of the data, or to take some real world data sets, generate some random ground truth probabilities. So for each cell, we can say the probability that it was going to be in the experimental condition or in the control condition, actually generate sample labels from this underlying probability distribution in step 2. We can then essentially calculate the density of each of the samples using MELD, and then compare the relative likelihood that we get from MELD to whatever this underlying ground truth was. And we can compare this using MELD versus a couple of other ways you might want to estimate the probability of a sample label, either just using like a k nearest neighbors classifier, saying like for each cell, what's the average of its neighbors in the ambient space, or averaging over the graph. And here I'm showing for each data set, we used like 200 different random seeds, different data sets, different kinds of signals, different levels of enrichment, different sizes of regions, and that the graph-based kernel density estimate that we use in MELD is more accurate at recovering this ground truth probability than sort of other ways you might want to estimate the density of the samples. We performed a very similar experiment for VFC, where instead of just having this smooth probability function, we actually create discrete regions of the data that have different probabilities of being observed in one sample or the other, and then can use something like the adjusted RAND score to determine how similar our sample labels are to the initial set of clusterings that we had. Great. So at this point, we've talked a little bit about a very simple experiment, just T cells, one cell type, two different kinds of conditions, no replicates, some simulated data. The next thing I'd really like to demonstrate is actually that MELD is really powerful for analysis of complex experimental designs comprising replicates and many, many different kinds of cells. And so the next data set that I introduce you to is a Crispr perturbation assay, where cells from embryonic zebrafish so that the one cell stage zebrafish embryos were injected with Cas9 and guide RNAs targeting either cordon or tyrosinase. Cordon being a developmental patterning gene, tyrosinase being it's a gene required for patterning of these, like, I think they're iridophores, like basically just pigmentation genes. So they're done in triplicate. So there's matched experiments. We've got replicate A, replicate B, and replicate C. And here I'm showing you a fate representation of the data. We can see that there's a lot of kinds of biological heterogeneity. In the published analysis, what they did was annotate 27 cell types. I'm just showing a couple of the labels here. So you have 27 different clusters that they then annotate based off of what kind of cell those clusters contain, and then compared fold change between them. But what we do is get actually this sample label and then produce some per cell estimate of the relative likelihood. And so, what I hope you can see pretty clearly here is that in regions where there's a lot of red, we see a lot of high relative likelihoods, meaning high likelihood of being observed after coordinate perturbation. And regions where there's a lot of blue, we see lower likelihoods. And to sort of get at the question that Samit was asking a little bit earlier, what do you do if you have multiple replicates or multiple samples? When we run MELD, we estimate this per replicate, so per sample. So what we do is take the cordon A and tyrosinase A, create the sample associated densities, and then normalize the likelihood across those two to get this one estimate of the likelihood of observing a cell in the cordon condition relative to the tyrosinase condition. And we do this 3 times for each replicate. So we do the density estimation once, do 3 kinds of normalizations. And what we can see actually is this gives us some estimate of the underlying the sort of variability in the response to the perturbation across replicates. So 2 examples I'm showing here, we're looking at adaxial cells in this small cluster, or cells from a notochord here. And if you see, the relative likelihood in sample A in adaxial cells is relatively high, but in replicate B, it's moderate, and then replicate C, it's a little bit lower. So there's variation here. And so you should think that because there's variation in this likelihood across data sets, you maybe trust the estimate of the effect a little bit less. We know that there's variability in this cell type. But take, on the other hand, cells like the notochord, where we actually see depletion in the cordon condition and replicate A, moderate depletion in B, and then strong depletion in C, we actually think, what I would be pretty confident saying the notochord is consistently depleted as a result of cordon loss of function in the zebrafish embryo. So we can then take the average across all of the replicates and get some mean estimate of the relative likelihoods. And then we can also get some estimate of the standard deviation, or measurements of variance or variation in the data, and sort of rank clusters or individual cells based off of how similar they are across replicates. I will just mention down the road, it would be really nice to be able to actually create some sort of statistical test based off of an understanding of the mean and variance. But we sort of thought about this for a while, and coming up with a robust alternative hypothesis and strategy for doing testing is not straightforward, and so we're sort of leaving that for a future research effort. Great. Okay, so we see for this data set, we get some estimate for every cell how likely it is to be observed after the cordon condition relative to the tyrosinase condition. To show you why this is meaningful to do for every cell, let's see, Smee just sent me a text message saying, how do you calculate mean if you have different number of cells in each replicate? And the answer is that if during the algorithm, the first step is to normalize the sample indicator vector, which gives you you sort of get equal weight before doing the kernel density estimate, right? The kernel density estimate has to sum to 1 across the full data set. And so this normalization is what takes account of that. If you have really huge imbalance, like 1 cell in sample A and 1,000 cells in sample B, your estimate of A is going to be really bad because you only have 1 observation, but you could still get a number out at the end. Back to this section, how we're going to use this relative likelihood on a per cell basis to give us some information about the effect of the perturbation. I'm showing you on the x-axis. So here again, each point is a cell. The x-axis is the cluster from the published annotation of the data set. And then the y-axis here is the relative likelihood of observing that cell in the cordon condition relative to the control. And the gray dot here is just the average relative likelihood. And so what you can see here for OP, you have to forgive me, I know these are really short. The full cluster names are very long and would be impossible to read on a slide. But OP is optic precursors. We see that there's, depletion. It's below, the average is below 0.5. There's almost no cells above it. And there's relatively little variation within this cluster across those cells. But if we now go and look at every one of the clusters used in the published analysis, effectively what you would do if you only had these cluster labels is you just get these gray circles out as your estimate of the effect of the perturbation. And so I want to highlight here that if you took an individual cluster like this tailbud pre-Semitic mesoderm cluster here, what we see is that it is overall enriched, which is what we expect. One of the things that's really well-known pattern in loss of function of choroidin is that the pre-Semitic mesoderm gets vastly expanded to the point where you can actually see this tissue growing much larger in the embryo than it would in a wild type. But if you look at the relative likelihood estimate, there's actually a really large variation within this cluster. There's actually some cells that are almost the most depleted cells in the data set. They're just a smaller fraction of this cluster. And so overall, the cluster is enriched. And this is actually really important, right? This one estimate, the fold change in abundance in your cluster, it hides an important biological phenomenon, which is that some cells are actually depleted. There are just fewer of those depleted cells. And so by looking at this, we can actually say, oh, this is interesting. We should go look at that cluster. There's probably different things happening there. So what we can do is actually just take those cells. So this is the tailbud pre-Semitic mesoderm cluster. The color here is just the same thing as the y-axis here. We're not rerunning MELD on this cluster. We're just using the original relative likelihood estimates. What you can see is that there's some, first of all, there's biological heterogeneity, right? We see there's multiple kinds of things happening here. It's not a blob like we saw with the T cell data set. And looking at this estimate of relative likelihood, we see some of those populations are depleted in the treatment condition, and some are enriched in the treatment condition. We can then use vertex frequency clustering to partition the data into different kinds of cells based off of not only the geometry, but also the fact that there are different responses to the perturbation, and identify subpopulations of this cluster that are transcriptionally distinct, functionally distinct, because we know what the cells are. They go on to become different kinds of cells and have a different response to the perturbation, which gives us a new increased resolution into the kind of experiment, the result of the experiment. And one of the things I really want to highlight here, what's really important, is that if you do traditional clustering, right, you have some measure of cell state, you do some partition of the data, and you say, you know what, this cluster size looks fine, I'm happy with this, and you maybe don't look necessarily, you don't have this access, so it's hard to identify this sort of histogram. What you typically do is just within each cluster, compare genes from the treatment condition and the control condition. But what you can do with vertex frequency clustering and MELD is actually take that cluster, separate it up into functionally distinct subclusters, or at least transcriptionally distinct subclusters, and now do comparisons between each of these subclusters to identify a gene signature. And this is really important because what we're seeing in real data is that transcriptionally clustering just based off of global transcription, just based off of the data set, sometimes means that you have subpopulations of cells with different responses, like some are enriched, some are depleted, some are unchanged in the treatment condition relative to control. And this now gives us a new way to identify gene signatures. And so if we go back and look at this tailbud pre-Semitic mesoderm cluster and the published analysis, sort of the standard way of analyzing this data set, you just get this bottom row here, which is you take all the cells from the cluster and you compare the treatment and control condition. Here we're looking at MyoD1, which is a marker of adaxial cells, which go on to become muscle precursors. On the right, we have TBX6, which is a marker of the pre-Semitic mesoderm, which goes on to become somites. And if you just look at expression differences between conditions, you'd say, a standard differential expression test would say there's a significantly differentially expressed, this gene is differentially expressed in that cluster. This population of cells expresses that gene more. But actually, if you cluster the data into these relevant cell populations, what you observe is that with small exceptions, there's no difference in expression of these genes as a result of the perturbation what you actually see is a difference in abundance of cell populations that are expressing those genes. And this is what's really important. It's not that MyoD1 is upregulated when you knock out CHD. What happens is that you see more cells that express MyoD1 after knocking out that gene. And this is something that you really only get when you use the clustering or partitioning of the data that is relevant to the response of the perturbation. And this is, I think, a really important finding for how we're going to quantify gene signatures, like what genes are upregulated when you perturb a data set in some way. And so with that, I maybe am going to just show one quick slide here. Sumit had asked about what happens if you have multiple kinds of perturbation or multiple conditions, more than two. And so here I'm going to show, this is a data set of pancreatic islet cells from our manuscript where we have 3 different donors in 2 conditions. So these are, I want to basically just get you from this slide is that these are beta cells, these are delta cells, these are alpha cells. But what we can do is actually, oh, that was not supposed to be there. What we can do is look at the, here, I'm just going to delete that. Look at the donor labels over the data set. So here we have 3 sets of donor labels, and then we can sort of normalize them independently. So we do this density estimate across all of them, we sum it all to 1, and here's you're seeing now 0.3 is the sort of equally likely to be seen in one condition versus the other. Red means enriched, blue means depleted, just gray now is at 0.3. So this is a little bit clearer. Let's see. So before I go to the conclusions, I also want to talk for just a couple of seconds about this single cell open problems project, which I think if there are people here who are interested in methods development, it would be really great. We're really excited to have some collaboration on this. I know this is going to be this will only be a couple of minutes here, but one of the projects, one of the things we have now, right? So this is what I just spent a lot of time talking about is that here are a bunch of methods that we've benchmarked, we think are really good, we did comparisons on, and are applicable to single cell data sets. But one of the problems, is that this relies on, me going out and doing these benchmarks on my own. I control the notebooks. It's not that easy for someone new to sort of add methods to this, sort of expand. They'd have to sort of do all this work all again by themselves. But one of the things that we sort of see in the sort of machine learning community is that there are a lot of open source, publicly accessible benchmarks. And so these benchmarks, there's a popular website called Papers with Code that maintains a state of the art for different kinds of tasks. You might break tasks down into like computer vision, where you want to do like image classification. Is this a dog? Is this a cat? You might want to segment an image. Like, is this part of the image a dog versus a cat? What's someone doing? And what we notice is that in machine learning, these kinds of benchmarks have formally defined tasks, like computationally, mathematically defined tasks, that easily accessible data sets, quantitative metrics, many benchmark methods, and that they're continuously updated. Someone does a new paper. Since we've worked on this, 2 new papers have come out doing this kind of analysis. And so they're continuously updated from one core source. And so there's been a really exciting effort that's been sponsored by the Chan Zuckerberg Initiative to actually create a publicly available benchmark like this, where you can actually see how different metrics perform against each other. And so if you can go to this website right now, openproblems.fyi.app. It's not perfect. We're still sort of in beta phase. We can actually create benchmarks that are updated based off of a GitHub repository. So you can have data sets that are available. If anyone is interested in learning more, I think I'm just going to say, please feel free to shoot me a message in the chat. Would love to talk more later.
You might want to just mention the types of open problems there are right now, just like 4 of them.
Yeah, so right now we're working on a number of different kinds of problems. One of them is around data imputation. We've got a task around dimensionality reduction for visualization, multimodal data integration. So say you have a site-seek and attack data and single-cell RNA, and you want to identify, which cells match between each other. There's data set on label projection, like annotating a new data set. Let's see, there's a few others that I'm sort of forgetting off the top of my head, but those are the kinds of tasks that we're really interested in working on, and we'd love to see new tasks be added sort of as time goes on. And they're actively working on this, and there's going to be sort of a remote hackathon at the end of March that people who are interested should contact me. We can get you on the invite list. Great. So in conclusion, what I talked to you about today, a bunch of different kinds of things, but the key heart of this section on perturbation analysis is a framework to quantify the effect of an experiment on a per cell basis. I showed you an algorithm called Vertex frequency clustering that groups cells not only based off of their transcriptional signatures, but also how they respond to a perturbation, which gives you more information about what kinds of cells they are. And I want to also maybe just make it clear that I showed you this for single cell data, but actually this method is applicable to anything where you can build a similarity graph. So if you remember, step one is you take the data set and you build a cell similarity graph. Anything where you can build that similarity graph, this metric is applicable to. That means that if you have attack-seek or site-seek data, that's perfectly fine. But you can also do this for like fMRI data. If you've got fMRI samples and you have connections between voxels, there's a lot of applications for this kind of graph-based density estimation to quantify differences between categorical variables. Moving forward, there's a number of things we really would like to do. So one of the things, imagine now you don't just have 2 or 3 samples, but you've got 10, and you want to know which 2 are the most similar to each other, which are the most different. One of the ways I think makes a really, sort of a mathematically robust way to do this is using a framework called optimal transport, which sort of takes our MELD probability distributions and calculates the distance between them. I mentioned a little bit before about maybe wanting to do some statistical testing. For each cell, you want to say, what's the probability that it was, actually enriched more than would be expected due to random chance. So that is adaxial cells I showed you that were high in one sample, moderate in another, low in a third. Those you might want to say are actually not significantly responsive in the same way as those notochord cells, which are always depleted across samples. There's some ways we could do sort of Monte Carlo testing to do that, but a little bit of work down the road. Right now, Vertex frequency clustering only works in practice on data sets of, let's say fewer than 5 or 10,000 cells. So if you have 50,000 cells, it doesn't work very well. What we recommend people do is just get coarse grained clusters first, and then use VFC as a secondary clustering metric. But one of the things we're actively working on is using wavelets to speed up VFC to make it easier to run this kind of clustering across larger data sets. And of course, one of the things we're always interested in is better ground truth. Splatter is a very restricted way to generate those quantitative comparisons. One of the parts of this open problems benchmarking is to do this better. With that, I just want to sort of thank everyone in the Crispr lab, especially my advisor, Smitha. I realize this is a little bit of an old picture, but anyways, so Smitha has been really helpful in guiding this project. Jay Stanley, who's now in the applied math department, has been really helpful in formulating the mathematical frameworks behind a lot of this, along with Alex Tong. David Van Dyke was, I think, one of the first people to really think about this kind of smoothing labels over a graph, and there's some interesting things we can do with that. Guy Wolf has been really helpful with leading some of the more mathematical sort of conception of the paper as a senior author, and Scott has been invaluable in working on the software and sort of discussions around the code. So at that, I really hope you all are left with is that we now have a framework to quantify the effect of experimental perturbations in single cell data. The manuscript is up on bioRxiv. We have a Python package available on GitHub that's fully documented with examples and a tutorial. And we just got proofs back today for our publication, which will hopefully be out in Nature Biotech in the next couple of months. Happy to take any remaining questions in the last few minutes. please feel free. Anyone who wants to unmute themselves and ask any questions, have any questions about your data set, method, computation methods in general, happy to.
Hi, thank you very much for the talk. It was really interesting. I was wondering, do you have any advice or method? You know that many users use different type of pipeline that are maybe not necessarily the same as yours. Like for example, if I mention Surat, how does it interface with this method? Which point should we transfer between the two to apply MELD?
Well, so I guess like it's a little bit hard to answer that question because unfortunately Surat is like 5 different kinds of methods and also a software package. And so if you're talking about like, this is in R and the Surat package is implemented in R. But there's also like Surat integration, there's Surat normalization, there's Surat clustering. So I guess like what I would just say is like anything that you would use as input to say like UMAP or FATE or your clustering algorithm, that sort of processed data set that's been, you know that there's no strong batch effect where you can build this kind of single cell graph. That's the matrix that you want to use as input to MELD. Does that make sense?
Yes. Yeah.
Yeah. And Samid is saying that he's used. Yeah. I mean, you give your single cell matrix, a single cell experiment object. There's like a counts matrix in there. It needs to be library size normalized, and we do square root transformation. But anything you would use as input to your visualization algorithm, you should use as input to MELD. Gabriel asked a question: can the likelihood be used to measure changes in cell type composition after a biological perturbation? I'm not sure, since a composition test may need a distinct / specific types of statistics. And so I guess what I want to say, so there's actually 2 separate questions in here. So measuring changes in cell type composition after biological perturbation is exactly what we're doing. It's an estimate. MELD is an estimate of the effect size at every individual cell. If you want to do a composition test, that requires a specific statistical model that has some assumptions about a proper alternate hypothesis. And I think that is the one that's really hard. Like, what is the proper null hypothesis for if there is or is not a meaningful change in abundance? What I had thought about doing is saying, we have now this relative likelihood over the graph with some probability, the average across all the replicates. Now, what if I use that to just generate new sample labels, like 1,000 times, right? Just do 1,000 samples. If a cell is 0.8 relative likelihood, that means that it's 80% likely to get the experimental label, 20% likely to get the control. You can just run this 1,000 times, get random samples, and then run the MELD pipeline and say, like, is that cell consistently enriched? Is it consistently at 0.8? Or is it because of some weird, sort of feature of the graph that it has high variation, if that makes sense? And Katie also just pasted my email in the chat. Please feel free to shoot me an email if you're interested, if you want to talk to me more. Any other questions? What are your other questions? There's 50 people still here. I'm sure some of you would like to know some more.
I have a question if I'm showing up.
Great. Yeah, please.
So I think this was a great talk, and I think you developed a really great tool that more rigorously looks at the effects of these perturbations, experimental perturbations. I'm curious though, and you sort of started to touch on it in this last comment about the sort of rigorous quantification of both technical and biological replicates of samples going into this pipeline. Yeah, just if you could sort of talk more about that and how you see this sort of moving toward that.
Can you clarify a little bit more? Like, can you give me an example of like a particular kind of data set and question that you?
Yeah, I mean, just even in some of the data sets you had, or if you're stimulating a set of cells with TNF alpha, you might do this experiment, 3 to 5 times with different, input cells that might also have some biological variability in the way that you've isolated them or passaged them. And at this point, I'm still relatively new to single cell RNA-seq, but there's not a lot to quantify or try to perform any sort of rigorous statistical comparison between technological and biological replicates. And yeah, so I think that that's sort of what I'm trying to get at, but from also, I admit, a very naive standpoint. So
no, it's a really good question that I think a lot of people have, and I think, you know, what I would say is that I'm not surprised. I think that this is a common question that people have when they're coming from sort of classic genomics, where there are pretty well-defined statistical tests for things like differential expression, where we think we actually have a pretty good idea about what the noise model is from different sequencing data and bulk measures, to single cell, where at the end of the day, we actually don't have any good statistical models. Like, I think a great example of this is that for about 3 or 4 years, there were dozens of papers talking about this phenomenon called dropout, and having this idea that some genes in some cells would randomly be 0. Like, statistically speaking, that was what the model assumed, that in some cell, you maybe have 10 UMIs, but some random thing happens and you get none of them. This is not biologically plausible. What we think is that every single transcript, every mRNA transcript, has some probability of the same, you know, within the same gene, there's some probability that you observe it, and you're not ever going to get 10 and then get none. What happens is each one has some probability, and maybe just because you're doing counts, you miss all of them, but it's not like it was set to 0 in that cell, which is what the statistical models would assume. So because of that, the statistical model, although it will very easily give you a p-value and say, this is the way I fit that model to this data, because of that, I can say that there's 0% chance that it's not differentially expressed. But if the model doesn't fit the underlying way the data was generated, the p-value is meaningless. And so instead, I think one of the things I heard in my first semester probability class here was, you know, everyone loves p-values. They want to know for sure that what they're seeing is real. But what's more important is an estimate of the effect size. You want to know is how high is it? And so what I would say is in your experiment, you have 5 estimates of your TNF alpha. You have 5 different replicates. And what I can say to you is that, you know, on average, that cell is 20% more likely to be observed in the TNF alpha condition relative to others. Or I could say, you know, but it's you know, out of all the cells in the data set, that cell is like the 90th most variable or something like that. You know, so you can get some of those estimates. Now, just, you know, then say, so if you were really wanting to go do a further validation experiment using like a true orthogonal, using like a true orthogonal biological measure, you might get a, you might not want to look at that kind of cell. Does that make sense? Does that answer your question?
Yeah, I think so. I mean, part of it is just like, as you mentioned, you can go through and, you know, randomly subsample your data set as you run the MELD pipeline to convince yourself that what you're seeing is real for, I expect what might be in some cases, like sort of controversial conclusions you're coming to with the frequency vortex clustering. But it seems like it might be hard to convince people with just, you know, a single set. And this is maybe just a problem of single cell to begin with, is that you can't just use that information to especially put forth sort of more controversial conclusions. So yeah, that answers my question.
Yeah, I mean, I think that to some extent, like, if you have a controversial conclusion, the burden is on doing more independent experiments, right? Because at the end of the day, I think single cell is much more useful as a hypothesis generating tool than it is as like a perfect validation. Like, if what you want to do is show that there's some subpopulation of cells that co-expresses some marker that you thought was previously mutually exclusive to each other, single cell is a great technology for that. If you want to show rigorously that perturbing this thing makes that population of cells not go down that pathway and instead go down that pathway, I really think it's important to have really rigorous, entirely unrelated experiments to validate that hypothesis. That's what I would say. I mean, I think that at the end of the day, that's one of the limitations, as you mentioned. I know we are also over time. I don't have anything immediately after this, so I'm happy to continue staying around and answering questions, of course. I guess I see Katie turned her video on. Do you need to give like some closing words before everyone goes?
Yeah, well, thank you so much, Dan. Well, feel free to stay if you have any questions. But as I said, Dan's email is in the chat. Feel free to email him if you have more. We can leave this open for 5 more minutes if people have more questions. Otherwise, we'll conclude today. Thank you so much, Dan. Thank you so much for everyone for attending this.
Yeah, it was really great. And like Katie said, I'm more than happy to stick around. Also, maybe we have a machine learning workshop where we teach some of these fundamental ideas about manifolds. It's full for the one starting next week, but we'll probably do another one in like May or something. Yeah. Any other questions?
Hi, Dan.
Hey.
Hi, I'm Tomo from David Toffler Lab.
Great.
I'm neurology. Nice to meet you. I have a quick question for you.
Yep.
So, you mentioned about the sample size if you apply this Kernel stuff with the higher dimension. What would be the ideal cell number of single cell RNA-seq data to apply this MELD accurately? Or
that's
the
like
result the other way, the result can be affected by the cell number?
Yeah. I mean, I think that at the end of the day, like estimating the proper number of cells for a single cell experiment for a particular biological hypothesis is an unsolved question. What I can tell you is that the T cell data set where we were able to identify a signature of TCR, upregulation or activation had, 5,000 cells in it. But the zebrafish data set was 35,000 cells. And I think, what I would say is like, single lane of the 10x chromium chip is 10,000 cells. I think that's good. I would try to target as close to that as possible. More cells, better.
Yeah, the reason why I'm asking about this sample number, cell number thing is sometimes the question that we wanted to, address is from very small minor population things from, within the data set.
Sure.
Yeah, so for example, sometimes you can find the very rare population with 100 cell number within the, 10,000 cells of 10x chromium.
Yeah.
Data set, but we wanted to compare that from healthy versus disease and those stuff. Yeah.
Yeah, I mean, at the end of the day, I think, the reason I was talking about doing this kind of like simulation for statistical testing is like, one of the things you see this population here, very, very small. One of the things that happens if you have a really small cell population is that the graph there is it's tightly connected within those cells, disconnected from the rest of the graph. And so small variations, in this small population, a difference of 10 cells having a different label can change your estimate of the likelihood by a large amount, right? 20, 30% or something. But in here, where there's a ton of cells, a lot of density, 10 cells being different is not going to change very much. At the end of the day, like, I cannot tell you what exactly is the right number. Again, I will just reiterate this, like, MELD will give you an estimate of the effect size. But I think you will probably find in that small population of 100 cells that the parameters that you use to build the graph are going to be very sensitive to the output. And so, like, I would want to be very delicate in how I analyze that data. And usually what I will say is that things that you find with MELD are identifiable or at least characterizable with another method. Looking at the data a slightly different way. You can validate it in a slightly different approach. One of the key advantages to MELD is that, there's so many possible things you could look at. This tells you exactly where to look.
Have you ever tried the downsampling to apply this MELD to see how many cells are, enough to have the consistent result?
I don't have the figure in this slide deck, but we did a downsampling experiment in the T cell data set, and I think we were still able to get 90% accuracy with this view as like 30% of the cells or something like that. It's one of the supplemental figures. I can actually probably find it if you just want to take a couple seconds here.
Okay.
I know
Good to know. Thank you.
Yeah, someone else asked about exactly that in the review process. That one or downsampling. So what I did here was I literally just took the T cells and removed some number of the cells and looked at the correlation between. So I guess this full data set was just say it was 5,000 cells or something like that. And so I think even within, and this is across like random draws, like there's, of course, as you go down, there's more variation, but like, I will say that you do start to get very, 1% of 5,000, right, is 50. So 90% or something. It's not terrible, but it's not as good. That's for sure.
Okay, great. Thank you.
All right, we're coming to an end. Thank
Perfect.
you, everyone. Thank you, Dan.
Yeah, absolutely. Thank you all for coming. It was a pleasure, and I'll talk to you all later.
Stop the recording. Thank you.